This is the third video in the EDEXCEL B3 revision tutorial series. Today we'll be looking at sex determination, sex-linked genetic disorders, and fertility treatments. So in this video we will use genetic crosses to explain sex determination in humans. We will look at why males are more likely to inherit sex-linked genetic disorders, and we will look at the pros and cons of different fertility treatments. So how is the sex of a person determined? Well, in the human body we have 22 pairs of matched chromosomes. However, we also have a 23rd pair labelled XX or XY. These are our sex chromosomes and our chromosome 23. All women have chromosomes X and X, whereas men have X and Y. When making sperm, the X and Y chromosomes are drawn apart in the first division in meiosis. This means there's a 50% chance each sperm cell gets an X chromosome and a 50% chance it gets a Y chromosome. The same thing happens when making egg cells, however, both egg cells will end up with an X chromosome. In order to examine the possible combinations of gametes, we can draw a simple genetic diagram. You will have looked at genetic diagrams previously, or you may have also known them as Punnett squares. They enable us to see the probability of getting a boy or a girl in this case. So we have the male chromosomes, we've got X and Y here. So here are the two combinations for sperm, and then for eggs we have X and X. So the first combination we can get is XX, and then we also have XX here. We then can also get XY as well as XY. This means that it's a 1-to-1 ratio between getting a boy and a girl. However, do remember that this is only showing the probability. So if you were to have 4 children, you couldn't guarantee that you would end up with 2 boys and 2 girls. You should remember genetic diagrams from B1. In B1, you will have looked at genetic disorders, including cystic fibrosis. However, some genetic disorders are found on the sex chromosomes. These are known as sex-linked genetic disorders. A characteristic is sex-linked if the allele that codes for it is located on either X or Y, so either of the sex chromosomes. The Y chromosome is much smaller than the X chromosome, so therefore it carries fewer genes, so most genes on the sex chromosomes are only carried on the X chromosomes. As men only have one X chromosome, they only have one allele for many sex-linked genes, due to only having that one X chromosome. Because men only have this one allele, the characteristic of this allele is shown even if it is recessive, because there is no allele to dominate over it. This means that men are more likely than women to show recessive characteristics for genes that are sex-linked. For GCSE, you need to know about 2 sex-linked genetic disorders. These are colour blindness and haemophilia. Haemophilia is a genetic disorder that only affects males and prevents blood from clotting. It is carried on the X chromosome, making it a sex-linked genetic disorders. Females, however, can be a carrier if they have one normal X and one haemophilia X. So one of the X chromosomes has that faulty haemophilia allele. Fertilised cells which end up with 2 haemophilia X chromosomes will not develop into a baby. The fetus is not sustainable. Colour blindness is another X-linked recessive disorder. The difference with colour blindness is that if you are a female who has both of the recessive alleles, this is an okay fetus, so therefore it will develop into a baby. We can draw a genetic cross for a sex-linked genetic disorder. So here we have an example for colour blindness. So as you can see, all of the X chromosomes now have an N after them. This is distinguishing whether the gene is faulty or whether we've got the completely fine allele. So we've got the capital N here for the dominant allele. This would mean that the person would not be colourblind. And then we have the recessive allele down here. So the recessive allele, which is only going to be shown if either both of the alleles present are recessive, or if the other allele isn't present in the case of the Y. So here we would have a non-colourblind male and a carrier female. So the carrier female wouldn't be colourblind herself, but we can see that she is carrying the faulty allele here. So putting together our genetic cross, we would have X big N, X big N, so this person would be female and would not be colourblind. We would have X big N Y, this would be a non-colourblind male. We would have X small n, X big N, so this person would be a non-colourblind carrier female. And then finally we have X small n Y, so it is this person down here who is going to be our colourblind male. So the chances of this couple having a colourblind child would be 1/4, so it would be a 1 in 4 chance. However, if the couple were to screen their child before birth and knew they were going to have a boy, then the chance would rise to 1 in 2, so 50% chance. Haemophilia is also an X-linked genetic disorder, so its genetic cross diagram would look exactly the same as this one for colourblindness. We can also show an X-linked recessive disorder as such. So we have our unaffected father and our carrier mother, giving rise to an unaffected son, an unaffected daughter, a carrier daughter, and an affected son. This is exactly the same as we were presented previously. However, we can then combine this information into a pedigree analysis in order to examine if a condition may in fact be sex-linked. We will now look at fertility treatments, bringing together what we have looked at in both this tutorial video and the previous tutorial video where we looked at the menstrual cycle. There are various different fertility treatments that are available to couples who can't conceive naturally. The first of these is the use of hormones. Hormones can be used if a woman has very low levels of FSH. Low levels of FSH prevent eggs from being matured. This means that no eggs are released and therefore the woman can't get pregnant. Both FSH and LH can be injected in order to stimulate egg release in the ovaries. The major positive to using hormone treatment is that it helps a lot of women to get pregnant when they previously couldn't. However, it does have some downsides. First of all, that it can be quite expensive as it doesn't always work, and also that too many eggs could be stimulated at the same time, resulting in unexpected multiple pregnancies, for example twins, triplets or even quadruplets. Egg donation involves women who can't produce eggs getting eggs from a donor and then using IVF in order to conceive. We will look more at IVF on the next page. As with hormone treatment, the major advantage of this is that it helps women get pregnant who couldn't before, this time because they can't produce eggs. Also, by using donated eggs, you can prevent the risk of passing on a genetic disorder from the mother because you've got a different genetic makeup. However, a major disadvantage is that it can often be emotionally difficult for the family knowing that the child has a different genetic mother. We can also use surrogate mothers, for example, what Phoebe did in Friends. A surrogate mother will be used if a woman can't become pregnant, then she may ask another woman to carry her baby for her. The woman who carries the baby is known as the surrogate mother. IVF is then used to produce an embryo, using either an egg and sperm from the couple, or can be used using egg donation or sperm donation. The embryo is then implanted into the surrogate mother's uterus. Then, after giving birth, the woman gives the baby to the couple who asked her to be their surrogate. So she gives birth to the child and then passes the child over to the designated parents. Again, a major positive of this is it allows a couple to have children if medical problems mean the mother can't become pregnant or it's too risky for her to give birth. However, a major disadvantage is that the surrogate mother is legally the mother of the child until it is adopted by the intended couple. The surrogate mother does have the right to keep the child, so she may decide she doesn't want to give it away after becoming emotionally attached to the child during both pregnancy and birth. The final fertility treatment that you need to know for your GCSEs is IVF. IVF stands for in vitro fertilization. In vitro means that it's done outside of the cell and is done inside a test tube or a petri dish. The opposite to in vitro is in vivo, meaning in life and therefore within the cell. IVF involves collecting eggs from the woman's ovaries and then fertilising them in a lab using the man's sperm. These are then grown into embryos inside petri dishes or test tubes. Once the embryos have developed into a small group of cells, a couple of them will be chosen to be transferred to the woman's uterus and womb. This improves the chance of pregnancy. Before the eggs are collected, the woman will be injected with FSH and LH in order to improve the collection of the eggs, as we will get release of more than one egg. IVF treatment has been around for about 40 years now, with the first woman to be born via IVF in the UK being Louise Brown, who was born in July 1978. And there have now been more than 5 million babies born in the UK via the use of IVF. IVF can also be used in order to counteract male infertility. If the man is unable to produce enough sperm, then a sample can be used and the egg can be fertilised in vitro. The major advantage to IVF is that fertility treatment like this can give an infertile couple a child, which is something that they couldn't achieve without it. However, there are some negatives as well. First of all, some women have a very strong reaction to the hormones, getting things such as vomiting, dehydration and abdominal pain. There have been mixed reports of an increased cancer risk from IVF, however, these risks are still being explored. And there can still be multiple births if more than one embryo grows into a baby. This can be risky, however, obviously the chances are slim. IVF is also very, very expensive, with most couples only being able to afford 1 or 2 rounds of IVF. And finally, some people are worried about the ethical implications of IVF and are concerned that couples might only want fertilised eggs with desirable qualities, so making designer babies. However, some people may see this designer aspect as a next step in our evolution, as we will be able to select against genetic disorders. However, it is very important to remember that all of these negatives will be far outweighed by the positive in the eyes of many, many people. As I have also mentioned, there have been over 5 million births via IVF treatments in the UK alone. So this shows that the negative side effects caused by hormones, as well as this potential increased risk of cancer, are slim chances to occur. This concludes this video, which has been EDEXCEL B3.3. In the next video, B3.4, we will look at the growth of microbes before moving on to look at both vaccination and immunisation in B3.5.